Hello!
Hi there.
Welcome welcome to Microbiology Journal Club, where we nerd out big about all things small. My
My name
name
is
is
Danny.
sorry.
Oh,
Oh gosh.
my name is Danny. In a previous life, I dropped out from my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC, dedicated aim to the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology, and I've mostly worked on bacteria, but since then I've worked as a research integrity specialist, and now I'm currently working as an editor for a scientific journal.
Every week, we meet to talk about microbiology, and today we're doing an overview of some of the coolest microbiology papers that we've seen this week.
And at the end of the month, we'll be selecting a paper to do a deep dive in. So make sure you message us at microtwjc if you see something that's caught your fancy.
You can follow along with any of the papers that we cover in our Shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us at microtwjc. So, boy, do we have a show for you today. First, we'll be looking at what SARS-CoV-2 does in the brain and how it gets past the blood-brain barrier. We'll also be looking at a tale of two mink farms and what happened when one of the workers came in infected with SARS-CoV-2. But even beyond SARS-CoV-2, there's even more infectious disease news. We might have figured out how a bacteria called Wolbachia prevents dengue fever, and
Mm-hmm.
also how neutrophil extracellular traps might make meningitis worse. And if you want to satisfy your inner bioengineer, we've got short stories about tangled up DNA and how that might be important for understanding how mutations happen, and how yeast factories can be programmed to produce complex biological products like isoflavonoids. So, whatever you do, don't switch away, and like, subscribe, and join, I guess, if that's an option. I don't know. Yeah.
Yeah, I mean, engage with us, right? We're in the YouTube chat. If you guys have questions, we're happy to answer them in the course of going through these papers.
Yeah.
And
So,
first up, we have uh, SARS-CoV-2 crosses the blood-brain barrier accompanied with basement membrane disruption without tight junction alterations.
yeah, now this is an interesting thing. So the blood-brain barrier is a big deal. It separates the blood from like the brain as
Hmm?
implied, and it's a really important thing to keep the brain as like an almost really protected site.
Yeah,
Um,
so even cells in our bodies have difficulty crossing the blood-brain barrier, right? Like people say that the brain has its own like immune cells because of this difficulty. Yeah, and it's really just trying to keep that particular area very safe from the outside world.
yeah, and this paper is interesting because it looks at a mouse model to investigate. Because during like various, because I think we've touched on this before in previous studies where SARS-CoV-2 does enter the brain.
Yeah,
People have, yeah, it's been seen there. It's been linked to many symptoms in that area. So the question is, how does it cross over
right.
from the blood into the brain? Um,
And I
and
think
this,
I think from what I remember of some of that stuff, they do think one of the ways that it was thought is it could go like retrograde on the neurons that innervate the olfactory section. But there's many ways into
yeah,
that, right? Depending on the cell types, um, there could be other ways in. So yeah, blood-brain barrier. That's like the the way, right, to get into the brain is is crossing that.
yeah. I think that's an important point because we did get that hamster study where they did these pictures of the hamster and they noticed that the nerves were getting infected with SARS-CoV-2. So
Hmm.
this also
Hmm.
looks in hamsters and in a mouse model, a mouse model which has the human ACE2 receptor, so
Yep.
it can get infected by SARS-CoV-2. And they managed to
Hmm.
infect the brain, and you get this like picture of the brain, but with these purple areas lit up where the SARS-CoV-2 is.
Oh
Um,
yeah, that's a lot
yeah,
actually.
that's yeah, that is quite a lot. So I mean, you can see that there's quite a lot in various areas, and when they zoomed in, they noticed that it was actually on these endothelial cells. So I think like the very circulatory system in the brain, the cells that was like surrounding that, like these things called brain microvascular endothelial cells,
Hmm.
are are like kind of they're part of things that make up the blood-brain barrier. So you can get like think vessels going through the brain, but these
Yep.
like kind of still form a barrier there that stop like a lot of the dangerous
Yeah,
stuff getting through, I think.
and I think that that's the importance in the title about the tight junctions, right? That
Yes.
part of what makes the blood-brain barrier a barrier is that there are specific cell-cell there's specific cell-cell connections that are relatively impermeable to cells, especially cells. They don't want like cells crossing that barrier, but even like certain compounds, right, have difficulty going through that. And so the only way to get through the blood-brain barrier is to be like to get into a cell, right? There's no nooks and crannies. The idea is that there's no nooks and crannies between cells to cross, right? You have to go through a cell. That's the intention, at least.
Yeah, and like what they found here is that they indeed like the SARS-CoV-2 does seem to be going through cells, and they do this through they look into animal models. They also create their own blood-brain barrier in vitro model where they take like a two-chambered like uh, well, and they add like the
Yeah.
basement membrane to one level, and then susceptible cells, and then they add SARS-CoV-2 to top to see whether SARS-CoV-2 can penetrate or not. So it's kind of neat that they do all these little like kind of ways to try and analyze how, and it seems that SARS-CoV-2 just can infect these cells, and they damage those cells, and that causes yeah, they and it causes, I guess, some leakage as well. Hold on, wait a minute, I might be jumping across to the next paper. Oh no,
Yeah.
they
Leakage
do
is
see
definitely.
leakage. In this, so they basically put in a dye in the brain of the mouse and
Mhm.
see like what happens in mice that are infected with SARS-CoV-2 versus mice that weren't, and they see this dye can spread around the brain a lot more quicker.
Mm.
Um, so
Yeah.
there's one like image where you can see like there's a lot of like dye that's concentrated in a specific area because there's been this breakdown in the blood-brain barrier because of SARS-CoV-2 damaging the cells
Hmm.
that are affected by it. And if I
Yeah.
recall correctly, because we've got like two very similar papers that are both really interesting. Uh,
Yeah, I
uh,
think that's yeah, that is very it's so in this one they're looking at two animal models and asking this question, right? Like can it get through that blood-brain barrier after seeing that it does, right? When they've infected the animal.
yeah,
Um and they show so it needs that ACE2, the receptor. And they also look at this metalloprotease.
yeah, matrix metalloproteases, which are quite important compounds. They tend to break down. They
They can break down the cell junctions.
Yeah, they're tools that the immune system uses to enter areas that it doesn't usually enter. So if there's a disease or some kind of like infection, immune cells will break down those areas and get in there and attack whatever disease is there. But of course,
Mhm.
this kind of thing can backfire a lot in really big infections, in which case it can cause a lot of damage just because the neutrophils or whatever is busting through all these different vital organs.
Yeah. Yeah, so it seems like in their model they say that it's matrix metalloprotease that's mediating the transduction through the
Mm.
cell. So something about that it could have so many different. It's hard to I don't really know about that cellular signaling pathway, but it is dependent, at least what they've shown here. Yeah.
Yeah,
But it doesn't break
but
down the tight junction. So it's like
yeah.
but it could kill the cells but it lets dye through. So there's it in what's happening.
I
Yeah.
think so. Yes.
Cool. And
Yeah.
that is quite
And
related to the second paper that we have on the docket, which is my computer froze. Uh,
the SARS-CoV-2 main protease Mpro causes microvascular brain pathology by cleaving NEMO in brain endothelial cells.
Thanks. Yeah, so Mpro, this is the protease of SARS-CoV-2. If people recall, like there's these open reading frames, and sometimes in a lot of viruses, when they translate from one of these open reading frames, it actually makes like a really long protein that has to get chopped up into the little pieces that actually
Mm.
do what they need to do. And so Mpro is the protease that does that, like the most important protease. But what they find in this paper is actually that protease also seems to cause some pathology when it's exposed to brain endothelial cells. So similarly to the paper before, we're talking about the action of SARS-CoV-2 on the endothelium in the brain.
Yeah. this also has some really cool. So it has some. So it stems from this like idea of made this observation in humans of these string what are they called
Yeah.
again? String vessels. Yes. So this is an interesting concept that when capillaries get damaged, they can eventually like die out and then leave these things behind called string vessels that don't have any like cells in them, but they just are these kind of remnants of previous like blood
So
vessels.
it's a
And
marker of pathology, right? And so
yeah,
this is something that you see it, you're like, oh, something's happened, right? There's some damage that's occurred in order to generate this particular structure. And what's nice in this paper is that they're showing that in some human samples. Yeah, where they
yes,
see, okay, in some, I think they're autopsies. Um,
yeah,
that oh yeah, like people with SARS-CoV-2 have more of this evidence that there's been some sort of microvascular damage.
yeah, definitely, and yeah,
And
that's
they so then in the paper they
and
try
again,
to discover why.
yeah,
And driven a lot by this idea that um, in order to create this damage, they must infect the cells that are right that are in this zone. And in order to infect those cells, they have to have the receptors and susceptibility to that. And we sort of know this before, although I don't think we've ever talked about BSG. Apparently, it's something that in the literature, it's another potential receptor for SARS-CoV-2. But we do know ACE2
yeah,
and we do NRP1. That's neuropilin.
yeah. BSG, I haven't heard about that one yet, but I think that might be something to look into.
Yeah.
Um, but yeah, again, they also work with hamsters and in a mouse model as well. They look at like the endothelial cells and they focus on parasites a little bit because those are also part of an important part of the blood-brain barrier.
Mhm.
Um, and they do some neat experiments. Let me see if I've got it right. They've got some. Yeah. Did I get the right paper on this, or is it some?
What, what's the experiment that you're
Oh,
remembering?
I'm remembering something from much further on. Sorry. Oh my gosh.
Yeah, so
I
in this one, they're essentially once they see that in humans, they replicate that formation in a mouse model. And
yeah.
specifically, there's this NEMO protein, which I don't know much about, but it's a protein that it's an immune
It,
protein that is inside of these endothelial cells, and it's being cleaved right in sort of it doesn't want to be cleaved except in very specific instances. But here, SARS-CoV-2, when it is able to infect these cells, it triggers this cell cascade that causes the pathology.
okay. NEMO stands. So you might know it actually by a different name, but NEMO stands for nuclear factor NF kappa B essential modulator.
Ah,
So NF kappa B is really really important protein for immunity, and this modulates that. So if SARS-CoV-2 cleaves this protein, it's a big
yeah,
deal for the signaling
yeah, for
because yeah,
immune
NF kappa
signaling.
B does a lot.
Yeah, so maybe
It's like
I was just gonna say
yeah.
maybe there is also like there is an evolutionary reason, right, to want to cleave some of these immune proteins, right? Like we always talk about these multifunctional proteins inside of viruses. So saying that the
Mm.
protease, which helps really make the functional proteins of SARS-CoV-2, also ends up cleaving this immunity protein, that, this could be this multifunctional thing that we always think about.
Yeah, and that kind of sets off a signaling cascade that causes those brain endothelial cells to damage themselves, essentially, to like break
Mhm.
down, and thus causing the blood-brain barrier to break down and allowing SARS-CoV-2 to go through, and causing a lot of that pathology that we've been hearing about related to SARS-CoV-2 in the
Yeah,
brain.
so I think in figure six, in figure six, they actually show like this is very similar to the tight junctions paper, or almost actually a little bit different because here the tight junctions are breaking down. Right? So
Yeah,
here they show a mechanism
yeah,
in which that it can directly break down those junctions and like seep through, right? The side. Yeah.
which is fascinating because they're both using pretty much the same models,
Yeah, yeah.
but they're coming to different conclusions.
Yeah, I don't. I feel like I haven't we haven't dived deep enough into these papers to really know like what's driving that. Is it maybe infective dose that they're changing up, or like something about the time point at which they're looking at? I feel like something like that has to be the difference between them.
Yeah,
Yeah.
I feel like they are looking in different
Hmm.
places. Like so, the latter paper was focusing a lot more on a specific cell type, whereas this one's delving a little bit more into like the molecular biology of it, and they're also paying a lot more attention to things. So the other paper might also have these string
Yeah.
cells, these but the string vessels, but this that they don't focus on that. They focus on something
Hmm.
else, and so I think they might be using different measurements, and because of that, they're coming at the problem in different
Hmm.
directions, and so we're almost seeing like multiple aspects of the same
Yeah.
thing.
And I mean, we don't, right? I think this is always the case when we talk about models, again, disease models, is that like
Mm.
when are these molecular events happening in the course of pathogenesis? In the model, we know
Yes.
we like you kind of like zero in on like that molecular chain of events, but you still have to you don't quite know where that relates specifically in the human disease, right? Where does that correlate? And
Mm.
so like they correlated the pathology, the endpoint, right? But
Mm.
like the steps that go up, like maybe first, I'm just like sort of spitballing, but like to try to synthesize these two pieces of information, maybe first SARS-CoV-2 travels through the cells, right, to get access, and then later on it starts breaking down, right? Like only when it's uncontrolled or something, right? It starts breaking down these junctions, it seeps in. But yeah, it's
Yeah.
I can't know, especially go not without
I
going
mean,
into
this is a
detail.
yeah. I mean, I think it could be quite interesting to analyze what the differences are, like in their
Hmm.
approaches, and then that might be an interesting idea because SARS-CoV-2 has got quite a diverse like symptom. It can affect so many different organ
Yeah.
systems, and it's got a wide kind of target
Yeah.
profile. So seeing how subtle differences in the way it arrives at a certain place can change that outcome could be very
And I think
interesting.
the other thing I want to say here is that, they have this endpoint where it's like, oh, damage, right? But like maybe in the course of a regular infection, this also like it's controlled after this, right? The damage happens, but people don't necessarily die from this damage. And this could be, I think that these types of studies are also trying to give us those mechanisms that we can start attributing to the long COVID symptoms and neurological side effects is like here are some damages that are happening now in the brain, so like can we correlate that or can we somehow relate that to clinical outcomes, and then that may give us this understanding, yeah, in that disease process.
Yeah, it might be. I mean, it might be helpful if people trying to find a biomarker for long
Mm.
COVID, even though like they're um because I guess it's really difficult research when you're looking at it from the kind of healthcare and social perspective, and people really want some kind of biomarker where they can say yes or no, even though it might actually be a continuum of different diseases that are all caused by SARS-CoV-2.
Hmm.
So it's quite
Yeah.
yeah, long COVID really scares me because it is just a really difficult, complex
Hmm.
disease.
I was gonna say, like, from the biomarker perspective, um, it's also like difficult to think in that context here because like this is brain tissue, right? That's being looked at. So we're not
Yeah,
gonna have a. This is like they had to do those post in the autopsy specimens to see that pathology. So I mean, but it could, again,
yeah,
knowing the mechanism now could give people this thing like, oh, actually, but when that breaks down, or like when these junctions break down, for example, maybe we'll see something come out in the blood, right? This is like directing the search, right? We never know what
yeah,
we're looking for in the world, like because there's just so many molecules that could be impacted by a disease. So studies like this that are, yeah, uh, zooming in on a specific model of a very specific moment in a infection, like narrows down that field and then can inform later hypotheses.
yeah.
Cool.
Right. I think we can move on to the next paper, which is quite an interesting story, actually. Outbreaks of SARS-CoV-2 in naturally infected mink farms impact transmission dynamics, genetic patterns, and environmental contamination.
Yeah. So this is like a old it's from old news, if you will. Like I think people remember because it was like such a excite I don't know if it's an exciting story, but like maybe like
Oh,
Worrisome story, thinking like, oh wow, they had to call all these mink farms because they got SARS-CoV-2. It was one of the first stories that we heard about transmission from humans into another animal, and it
yeah.
set up this idea that for scientists and public health officials that there could be reservoirs in certain animals that live closely with us, and
Mm.
that would be the gateway into like recurrent disease, something like that. Because I mean, again, SARS-CoV-2 doesn't mutate as fast as the flu, but the flu does this, right, in terms of it infects the respiratory
Mm.
tracts of or the gastrointestinal tracts of birds, right, and that serves as a reservoir for it to sort of seasonally pass through our population. The worry is that in these in instances where humans are passing it back into some sort of animal population, that just serves as a place where it'll stick around, right? And maybe get transmitted amongst those animals and then someday jump back into humans. So there's interest in
Yeah.
understanding exactly the extent of the infection in these types of animals and how does it spread in the facilities in order to make sure that these facilities can be designed or managed in a way that prevents spread between these potential reservoirs and humans.
Yeah. It's uh yeah. I mean, they go into some depth in this paper. So you've got like the dates of the
Yes.
outbreaks. You've also got uh floor plans for all these mink farms.
Yep. And this is
Um.
recent too. So if you're like, I feel like
Yeah.
the original, Mink news when it like gripped everybody was like very early in the pandemic.
Oh,
Yeah.
yeah. Very, very early. And again, I think that it was like one of the first hints of like, oh, SARS-CoV-2, what you might not be able to stop it from getting
Yeah.
endemic. It just might,
I
yeah.
actually just remembered. I think that wasn't the news article like minks rising from the grave or something like that.
Yes, yes, I do remember that. Horrifying. Happy Halloween, everybody. Happy
And it was just
Halloween.
because
Spooktober
it
is
was this
here.
mass culling of the minks and they had to be buried, but then, small animal bodies get bloated with gases and they came back up.
And yeah,
And
yeah, Mink rise. Yeah, like I literally feel like the headline writer just was grinning from ear to ear when like, okay, dead mink infected with SARS-CoV-2 rising out.
yeah.
Yeah, it was a horrible time was had all around with that.
But this is like, if you look at this timeline, this is 2020 to 2021. So like, there's an ongoing, right, an ongoing story of like mink farms. There are many mink farms. People want fur coats,
Yeah,
and it's ongoing in terms of the way this industry has to respond to the ongoing
yeah,
SARS-CoV-2 pandemic.
yeah. I mean, this is a paper that like does tell you a lot about the mink
Hmm.
industry, and ooh, not buying a mink coat in the future as far as I'm concerned. But
Yeah, I
from
mean,
the scientific
yeah,
perspective, yeah,
it's not for
no,
everybody. I think, yeah, people, it is. Yeah, we are just using animals for fashion in this sense, but yeah, you get this sense of
I
this
mean,
is the like they have a rearing period for these animals,
mm,
and that 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 correlates with the way in which people were vaccinated. And so there's you
mm.
get this idea of like the population swell within the farms, and yeah, who's these workers working in close proximity with these animals. Oh yeah, they map it out here. um,
Yeah, they've got. So there are two farms. There's Farm A, which is
mhm.
massive. So this is like a much bigger farm, and then Farm B, which is a bit smaller. So like it's. I mean, I would not be comfortable putting down floor plans for places like this because of security concerns.
Mhm.
But yeah, this is. And yeah, like I'm not. I don't feel good about how big those cages are.
Yeah.
Um,
This is interesting. I feel like
but then
that
yeah.
insight that you just said, like you wouldn't feel comfortable publishing it. Like I know this about animal research, and particularly in Europe, there is this idea that like you got to hide the animal facilities because some people like who don't agree with it, they want to take direct action against these facilities.
Yeah. Yeah, no, there is that concern, and I presume that the same thing is for certain types of farming as
Mhm.
well. Um,
but they've been open here for
So
whatever reason. Maybe because
yeah,
I think they want to show like a really clear instance for right people who run these types of facilities and like where those I guess like where those close quarters events happen, right? Like part of
mm,
seeing the facility like that is to get this sense of like, okay, here's where the humans are closely interacting, and then because these animals are living in this type of context, then they'll transmit amongst themselves once that initial leap happens.
yeah, and this is just the graph of the time course of the outbreaks for both of the farms.
Hmm.
They're somewhat different because I think one of the farms try to do a lot more isolating of,
Hmm.
and you can see like there's a lot fewer dead minks in Farm B compared with Farm
Yeah,
A. Farm A, just you got this
that's just a
bell curve
that's just
where
a classic like running through the infection curve, right? Like it goes up, and then now the population can't sustain transmission anymore, and you get it goes down.
yeah. Whereas I think Farm B's outbreak happened a bit later, so they did a few more controls and they did molecular diagnosis a lot earlier, and yeah, so I
Yeah.
so like I guess one of them is herd immunity, one of them is just
Yeah, test and
is
trace.
doing
Yeah,
isolation.
yeah. And they do cover again this burning question I think people have is like, what are the mutations that happen in order to get into the minks? And it's
Yes.
sort of right, like what are those mutations? Because they might tell us something about what like if you can then test those mutations in like a human animal model, right, and get the sense of what is or isn't possible, then you also have this idea of how difficult it would be for the SARS-CoV-2 that jumps into the minks to jump back into humans. But I don't, yeah, but
Right.
I'm not really sure what the entire what the takeaway would be, just seeing like the list of mutations here.
Yeah, I mean it's like a 95, like I think was it a 10% death rate for minks in like depending on which farm they went to. Um, but yeah, it was yeah. So this is a pretty interesting paper for just looking at kind of almost like a small model of like an outbreak and seeing like how that works and comparing it to our own experiences with dealing with these outbreaks.
Yeah, yeah. I mean, it's quite interesting in that respect too. Yeah, just seeing what goes into managing something like this, and the fact that they have those two farms to compare gives us some insight into like the
Yeah,
interventions that might be more or less useful.
yeah. Now, next paper is the replication and single cycle delivery of SARS-CoV-2 replicons. So this is more of a tool set for people who are in basic research who want to work with SARS-CoV-2 but don't want to actually have to deal with a dangerous
Hmm.
virus. So one way to do this is to chop up the SARS-CoV-2 genome and put it in different places so that it can't form like a virus that can cause a full infection. Um, so there's the idea here
And
was that they'd take
that's called a replicon, right?
yeah, that's called a
Yeah.
replicon. So a replicon is the SARS-CoV-2 like genome, but it's missing key, like in this case, the spike protein,
Hmm.
so it can't cause an infection, which is quite useful for us as
Yeah.
researchers. Um, and they create it in a way using like some special yeast technology, which is let me see, transformation assisted recombination, which is like essentially like getting everything together in one go. So it's got these. So yeast has these recombinases that aren't necessarily very fussy, so they'll fuse together genes that even if they're not very similar, so
Hmm.
it's a great way to try and put together a fancy like
Yeah.
genome. Um,
So they essentially
the
assemble like this spike deficient genome inside of a yeast cell.
Yeah,
Yeah.
and then use the yeast to express it, and also to yeah, they use the yeast to create cDNAs, which they then use to make the RNAs, and they infect those RNAs into cells that can have all the kind of replication bits and create like SARS-CoV-2 variants that can't actually cause an infection. They're just like balls that don't have the spike.
Yeah.
And what they can do to, and if you actually really want to model that infectious portion of it, so the great thing about the replicon is you can look at all the metabolism. So you want to test an antiviral, it's a good way to test it because antivirals tend to work against the RNA-dependent
Yeah.
polymerase, and that's so that's still active and fine. So you can still test your drugs against it without any risk of getting infected by SARS-CoV-2. You can also create a single-use SARS-CoV-2 protein where the host cell produces a spike protein, and that gets incorporated into the SARS virion, and then that can cause one infection. But so it can infect a cell, but since it doesn't have any of that blueprint to create a spike protein, it's stuck there.
Yeah, that's interesting. And do they do that? They do that with VSV, right? And this
Yes.
essentially it's a pseudotyped, a one-time pseudotyped virus.
Yeah, I like these sorts of tools where they try to like democratize science a little bit, trying to make things simpler. So you don't need to have a category 3 lab where you have to wear these giant suits and.
Yeah, so
Uh,
yeah, I think what you were saying, the utility being that what's happening inside of once the yeast, once you put the RNAs or whatever into the target cell that you want to infect, right, that were produced by this
mm,
in this technique, you get a cycle of replication that is the SARS-CoV-2 cycle of replication. Yeah.
yeah.
And so and they have a reporter in here too, right? They have like a fluorescent signal so you can see.
Yes, they have Gaussian luciferase, which is a really cool reporter because it's one of the brightest reporters out there.
Mhm.
So I mean, because I did my PhD in like with luminescence, so we're comparing promoters, and Gaussian is one of those things that it's a very fast reaction as well.
Mhm.
So that can be quite useful in getting the timings of things because sometimes luminescence reactions can be a little bit delayed. But anyway, that's just a fancy thing about that
Yeah,
technology.
but we had talked about earlier, I think maybe a few weeks ago, we talked about some news where they made the SARS-CoV-2 particles. They got a reporter, right? It had to be like a really
Yeah.
tiny one because they didn't want to break anything else. But here, the focus was a bit different. They're like, okay, well, we don't want the infectious particles, right? So they could just remove the spike protein and give it like
Yeah.
a more useful, maybe, right? A more tuned
Yeah.
reporter. And like, you don't get to see the infection dynamics, but what you do see is the replication dynamics. And even just
Mm.
seeing that one step, that one cycle of replication, you can test things like what's going to block that, right? I think that that's like,
Yeah.
it's an interesting, I didn't really think about this way of using, right, SARS-CoV-2 to do research, but it makes a lot of
Mm.
sense from the biosafety perspective, right? And trying to get more companies, I guess, in some ways, right, able to look for drug candidates.
Yeah, and there are all these like SARS-CoV-2 programs where we don't necessarily know what they do,
Hmm.
and this is an opportunity. And at the moment, they might not do things that are so a lot of the ones we're focusing on are tend to be the ones that are like essential for infection. But there's all these other parts of the SARS-CoV-2 life cycle that we don't have full clarity on, and this will give the opportunities for other people who want to study those specifically to have more tools.
Yeah, more people can study SARS-CoV-2, right? If there's some specific non-structural protein and there's an interesting immune interaction, you can study that in isolation from worrying about infectious particles at the end of the day. And instead, you have this proxy of like just butted off things with the reporter, expressing reporter.
Yeah, and that brings us, I think, to our next paper, which is a really interesting one. Targeting the coronavirus nucleocapsid protein through GSK3 inhibition.
Hmm. Yeah, so in this one, they talk, and I didn't realize this, but the coronavirus nucleocapsid protein has to get phosphorylated inside of the host cell by
Hmm.
host phosphorylase, um, and that
Yeah.
host protein is called GSK3. And what they find out is that if they inhibit GSK3, then they don't get as productive. The nucleocapsid protein isn't able to, I guess, come together properly, and they don't get really productive infection. And what is interesting about this paper is that GSK3 is inhibited in a clinical sense for neurologic issues through lithium. And so
Yeah.
then they go and they show that when you look back, sort of with this insight, that people who are treated with lithium may have better outcomes because they're not going to have as productive of an infection. You find that yes, indeed, that is the case. People who are on lithium and who happen to get SARS-CoV-2 um, had fewer of them were SARS-CoV-2 positive, I think is what they find out.
Yeah, I think this is an interesting study because I mean GSK3 is quite an important because it's one of those it helps control glycogen synthesis, and glycogen is a very important molecule because that's how we store glucose. So this affects quite a lot of really important like cellular
Metabolism, yeah.
factors. Yeah,
It's a very central protein.
yeah. Which is why, like, if you're taking a drug like lithium, some of the side effects can be things like weight gain because it affects your whole metabolism,
Hmm.
and so it can change. So yeah, it's I mean, this is an interesting study because they start off from this cellular perspective of okay, we know GSK3, there might be a connection here. Let's go through it and pick apart because there are two like flavors of GSK3. So which
Hmm.
one is it, alpha GSK3 alpha or beta? And they go through this like I mean, it's still very cellular studies, but so I mean,
I
I'm not gonna go to
I was gonna say,
so
I got the sense, like, to me, when I saw this paper, I get the sense that they maybe did that lit search first, but
yeah,
to get to this particular hypothesis instead of right, and then they followed up with all this biochemistry to be like, okay,
yeah,
let's show now with biochemistry that this GSK3 is having an effect on SARS-CoV-2. But I would assume that the first thing yeah, that's just my thought, based on how the
yeah.
story
I mean
went.
that's
Yeah.
Yeah, I mean that makes sense because I mean from a story perspective, it would be more fun if they did a literature search afterwards. Because again, retrospective like observational studies aren't necessarily the strongest form of evidence.
Hmm. Hmm.
Um, because uh, so I, but then
That's
again,
my
if it's
sense
from
of
the other
just
perspective,
looking at the
yeah,
figures and how it comes together, because it is interesting to think, or it's possible that people just have like this sort of intense knowledge of what's going on inside of a cell and say like, oh, like we know that SARS-CoV-2 N protein needs to be phosphorylated by something, right? And then try to like
mm,
pare down from all these things. It sort of makes my sense. Yeah, I mean, it says, oh, I'm trying to see in the last paragraph of the introduction, it does say that like, yeah, they're summarizing some of the things that they saw. Yeah, like they do see that there were these observations that things that impair GSK might impair SARS-CoV-2. So they have this sense that,
mm,
yeah, it could be there. It's funny to link it back to something that's already being given, right? In some ways, it's a natural experiment, right, that was being done,
yeah.
that they're highlighting.
So yeah, it's interesting. I'd be interested to see like further research on this.
I,
But I'm probably not gonna buy lithium yet
Yes, I
just
was
for
gonna say,
this.
yeah, I was gonna say the one thing that I think we should say here is that, right, like it is people do broad searches trying to find host factors that, inhibit a productive infection by SARS-CoV-2. But you don't always want to inhibit those host factors, right? There's a slew of side effects that happen, right, when you inhibit certain host factors because you need those host factors for regular life. And so like this is one of those things where it's like, yeah, there seems to be an effect, but would it be something that's well tolerated in a lot of different people? Like this might just be more like a last. These are maybe things that end up being like last ditch efforts, right? Off-label uses of drugs,
Hmm.
that when doctors like need some very specific option, right, in their circumstance, like they can bring to bear. But it may not be something
Yeah.
that, goes through like phase 1, 2, and 3 trials in order to relabel a drug for this use.
Yeah, there's a lot of stuff here, and in cells, I mean, there's a lot of different factors that can affect a cell culture that don't necessarily apply to humans.
Mm.
Um,
Right. And what we're
and
seeing here is the evidence is being done in cell culture, but they do try to link it right back to some population of people that are taking it out there in the real world.
yeah, yeah, they're both kind of not the. I mean, it's odd because I don't want to like say anything, but because people are uncomfortable with saying like with the people usually think of true and false. Like, does it work, true or false? But in science, that dichotomy is very fuzzy
Mm.
because there are levels of things we know, and at the moment it looks positive, but that doesn't mean that it actually will work in practice because we've got cell culture and observational studies, which are very they seem like they're saying telling a story, but there's always like an asterisk that you have to have in your head when you're reading something like that. That okay, let's not get our hopes up too much.
I think no,
there are
that's a
more
really
questions need to be
that's a
asked.
really good point. Like maybe we've talked about observational studies by themselves, right? And being like, there's all these caveats. We don't know like what are the covariates, how to do right? Like maybe there's things that assort those groups, not just what they have studied, right? The fact that one's taking lithium versus others who aren't. There's other factors that determine that difference, right, in SARS-CoV-2 susceptibility. So we've talked about that in isolation, like there's like issues with that. And we've talked about cell culture in isolation, right? Because it's like those are cells outside the body, right? And like they don't have the same signals. And so this paper is telling a story with those two things together, and it does make it more compelling, right? Because you have two things that correlate with each other, but it's still they both each have this major caveat. And so we would, of course, want to see, right, a more definitive way of answering this question. Is GSK3 inhibition a good intervention, right? Good clinical intervention for SARS-CoV-2?
Yeah, I'm afraid I have to say the dreaded more research is needed. more funding, please.
The classic refrain.
Yeah. And now next paper, we're moving away from the SARS-CoV-2 era and of our work, and now we're going into the more infectious disease route. So this one is novel symbiotic genome-scale model reveals Wolbachia's Arboviral pathogen blocking mechanism in Aedes aegypti.
So Aedes aegypti, that is a mosquito. And Arbovirus, that is a whole slew of viruses, including dengue, which is just a virus that's transmitted by mosquito bites, and we don't want those. And actually, if people have been following the news, I think arboviruses may also include Zika and West Nile. I think they may be in there.
Yep, that's right.
So, right, those are very high profile, right, viral diseases that we have an interest in disrupting. And what has been known for a long time, and actually is a strategy that's being used, is that when you infect these mosquitoes with Wolbachia, which is bacterial endosymbiote, symbiotic bacteria, it reduces the ability of those mosquitoes to transmit the virus. And it's a big question why. And the way that they're going about it in this particular paper is by modeling the metabolism of the mosquito and the Wolbachia, and showing that when they come together, there's some pressures put on the metabolism of the mosquito that makes them less likely or less able to replicate high amounts of this virus. First of all, I think this is a very strange mechanism. I've never experienced, and I am probably very poorly equipped to understand like how this work was exactly done in terms of the metabolic modeling. It might help to explain that metabolism is all of the chemical transformations that happen within our bodies, and we know a lot about the different enzymes that manipulate metabolism, and we know that also like when you eat certain things, right, like you get metabolites that have to transfer through your gut, right, and that gives you like a flux, right? You'll get more of this metabolite, which makes certain enzymes have to work harder, and somewhere along the way there are different choke points, right, that can slow down the flux of different molecules to where they need to go. And that's essentially what they've tried to capture here in this graph. Is all of those reactions. It's in a computer. There's a GitHub associated with this. Right? This is not, someone didn't draw this out by hand and figured this out. They instead put this into like a computer simulation. So the computer is simulating the effect, right, of a mosquito's metabolism, and they're able to like turn the different knobs to be like, well, what if they got a little bit more of this molecule, right, or a little bit less of this molecule? How would that change the amounts of other molecules in the mosquito? And specifically, what about certain molecules that we know are required for viral replication?
Mm.
Yeah.
Yeah, it's an interesting perspective because I tend to think that Wolbachia was almost like enhancing the immune system of these mosquitoes, but this paper actually turns that aside and focuses on actually like this competition that's happening between a bacteria and a virus for resources.
Yeah, yeah. I agree that I thought I would think, and they're not mutually exclusive, right? An effect on
Yeah,
the immune system and an effect in terms of competition for resources. Both of the things could be contributing to Wolbachia's mechanism of reducing arboviral infections. But yeah,
but
they focus here specifically in reconstructing this particular this scenario in silico and then testing. And they do verify, I think, that their model first works in different circumstances. They sort of, I think they have cell culture data from mosquito cell cultures that say like, yeah, like when mosquitoes experience this stress, their metabolism changes in this way. Does the model respond in the same way? Does this piece of code respond in that same way? And they show that it does, and that's what gives them the confidence to say that what we're showing in this model, right, by like nudging the knobs as if Wolbachia was changing the metabolism, that is realistic, right?
yeah.
Mm-hmm.
Like I think they link back to this idea that Wolbachia is gobbling up a load of cholesterol, and cholesterol is an essential entry point for uh, the arbovirus for dengue, or
Mm-hmm.
and also like take up all the amino acids that the dengue would usually be doing. So I think it's yeah,
Yeah.
it's quite an interesting way of answering the problem by basically simulating that and putting it on the computer. Goes okay, what happens if this Wolbachia is gobbling up all the resources? What does that mean for the dengue virus? And we don't it's weird because viruses do use up resources. We don't necessarily think of them like that. We tend to
Mm-hmm.
think of them as these unliving things that don't do anything.
Yeah.
But actually, when a cell is infected, they do have a metabolism. They are kind of alive.
Yeah.
So it's
I think one of
yeah,
the things that I like about this is that it's very so they're focusing very specifically. Yeah, first of all, very strange mechanism of in silico testing. But
yeah,
and they're focusing on this idea of like a transmission event, but it's in the context of this ecology essentially, right? Of like
mm,
these interaction with bacteria, and so I kind of like it. And we also, I think we talked about maybe last week or 2 weeks ago, we said that like there was something where they said like feeding sugars to mosquitoes like also changes their
yes,
infectivity in terms of
yeah,
arbovirus. So like there's a link here, right? Like there's other data that supports this idea that like stuff in the gut, right, does feed back into the ability for viruses to do what they need to do in their life cycle. And what do I want to say? Like, I guess it's just it's highlighting that fact, right, and showing this in another way. By itself, I don't think that we're fresh off of like sort of putting doubt on types of research data. Like by itself, I feel like this paper is like there's much to follow up on, right? In terms of,
yeah,
right? But it's kind of cool because they also have this tool now where they have like a modeled mosquito metabolism, and you could potentially ask other questions there, right? About mosquito biology if it's interesting to you, right? And how it impacts their metabolism.
yeah. I think you're right about that. It's just very much a like hypothesis generating activity
Yeah.
rather than it raises questions that can be tested later.
Yes,
Um,
yes, yes. I think that's very true
but yeah,
for a lot of in silico work, or that's the way I view in
mm,
silico work. It's like, yeah, you find out these amazing things like computers can put together, like can balance all of these different parameters, but then you want to make sure that it like so even if the model was validated by data, which this one was,
mm,
right, presumably, then you still have to take that and revalidate the specific finding that you pulled out inside of a physical system.
yeah,
Yeah, experimental system, I guess. Physical experimental system.
yeah. And I think we've said everything we needed to say on this one.
Mm-hmm.
So I'm going to move on to the next paper, which is one that I've been so excited about. I've been talking about it in the other papers, but um, DNA's treatment prevents DNA's treatment prevents cerebrospinal fluid block in early experimental pneumococcal meningitis.
Mm.
So this paper is about neutrophil extracellular traps. So we've talked about them a little bit, but essentially the idea is that neutrophils, when they're fighting against pathogens, can throw out this like net that can grab them up, and the net has like all this. The net has a lot of bioactive compounds that attack the bacteria directly, and it's wrapped up in DNA as well and structurally. So they're really interesting, like kind of so these things called neutrophils, they undergo netosis and they almost suicide kill a bunch of bacteria.
Yep.
But this paper
And I think
focuses on why that might.
I'm sorry, I just want to say,
Sorry?
I think that one time you brought us a paper about nets but they might also not die. There are some
Yeah, that's
neutrophils
right.
that may also just like somehow stick around, even they've like divulged all their nucleus or whatever, but they just stick around for a little bit more to gobble up some last things.
Yeah. Yeah. So they really complicated, really interesting. Um
Yeah.
but this paper focuses on why that might not be a good thing, how their activity might be a bad thing, and in the context of meningitis. So in meningitis, there's a lot of fluid buildup. So in the brain, there's this thing called the glymphatic system, which I learned about this morning,
Yep.
which is like the lymphatic system, but it's in like the brain where it kind of drains off a lot of the crap that happens that brain cells give off sometimes, like all the waste products.
Yeah.
And of course, the natural thing for that is that if the immune system plays a role in like looking at those waste products to see if there's anything dangerous in there, and if bacteria get in there, then obviously that provokes an immune response and provokes things like neutrophil extracellular traps to
Be sprung.
like to get sprung.
Yeah.
And so they do that. They have this model of meningitis where they inject some bacteria in the brain of some rats, and then they look and see what happens in terms of inflammation. And they got these images of the rat brains and these neutrophils forming their extracellular traps around the kind of glymphatic vessels.
Yeah. They're really nice images, and I like the
yeah.
they show you like the little atlas of the brain section that the close-up images are taken from.
Yes, let me zoom in. Yeah, you've got like a very much like a timeline of what's happening. You've got like a little atlas in the corner showing which part of the brain they're looking at.
Yeah.
And yeah, you can see these. You've even got like 3D images of like how these nets are forming around the brain.
Yes.
It's really, I mean, because there aren't many papers that take like images. I think this is the first paper I've seen where I've seen nets actually inside their like in situ in vivo.
Yeah.
So that alone is quite cool. But what they also find is that when nets are more active, there's more like fluid buildup, there's more like negative things that happen.
Yeah.
And they come up with this theory that the nets are actually blocking up the glymphatic system.
Yeah. It's a very plumbing-based hypothesis.
Yeah. They test it in a very plumbing-based way where they add a dye and they put it into like the brain of these mice and see where it filters through.
Yeah.
And see whether it can go into certain areas. And what they find is that if there is no like infection, the dye can travel all across the brain quite freely, but where there is an infection, it's blocked off from certain places.
Yeah.
And they even like look at the draining lymph nodes because we've got like these lymph nodes in the neck that you feel when you're sick. That drains off pretty much most of the head. So they look to see whether the dye shows up in those lymph nodes as well, because if the lymphatic system is blocked up, then that dye won't go in there when it should. And they find that the mouse lymph nodes, when there's like infection, are less brightly colored with that dye compared to the control.
Yeah, that's so interesting. I mean, again, this is I think nets are I think quite a new meaning like like 10 years, 15 years or something like that. But still like a very new mechanism in which the immune system acts, and there's lots that we're learning about it. It is very interesting to hear in this story that you found that that just like all immune things, there's always a good and a bad side, right? Like the immune system, like it needs to act in a very specific scenario in a very specific way to have its maximum effect. And in some edge cases, or maybe just we'll call them edge cases, but really they're just like maybe like in some cases, right, that the immune response is not appropriate or creates other issues in addition to doing their immune function, right? So here it's driving potentially some pathology, right? Not letting the the liquids of the brain circulate properly.
Yeah, I mean, yeah, nets are so new that I literally, I remember I didn't quite believe in them for like quite a while because they were so new. The assumption was, well, maybe there's just some kind of experimental thing because a lot of the early papers were just like ex vivo outside, so
Right.
maybe, and then
Yeah,
yeah,
it's you could.
and now
I feel
I'm
like you
like,
could imagine, especially when you think about it, like, oh, a cell dies and spits out all this stuff that traps bacteria. It's like,
yeah,
why? That's just normal. Like cells blow up and then stuff gets out, and sometimes it's sticky. But like more and more, I
yeah,
think people are honing down on the fact that this is a behavior of neutrophils, certain neutrophils, right? That like there are instances where they want to explode in this way in order to trap bacteria and yeah.
yeah. But now today I like, yep, I believe
Yeah,
now.
you
I'm a
believe.
believer in.
You believe, and also that it's not always a good thing, right? That there's
Mm,
possible that when the nets are deployed, it's not working for a certain scenario, and this is like a big theme I think that we always are talking about when we talk about the immune system. Yes, we want to make the immune system stronger and better at like defeating its enemies, but there are also actions that it takes that drive pathology, and
yeah,
yeah, so that's what makes I think some medicine very complicated, but also makes people super excited about like immunotherapies and like all these different therapeutics that might help us or help train and nudge the immune system into better outcomes.
yeah. Speaking of better outcomes, what they did after this was they figured out, well, if the nets are causing a problem, maybe we can figure out a better solution.
Hmm.
And think about nets, because bacteria have been fighting against nets for a long time. And one of the things that was the main line of evidence for nets' existence was the fact that bacteria produce these things, could produce DNases
Hmm.
that could dissolve nets and inactivate them
Yeah.
theoretically, but now we know it's actually true.
Yes.
Um, and so they go, okay, well, how about we take a tool out of the bacteria's book? How about we give some DNase treatment to these mice
Hmm.
to break down these nets? And they did that,
Hmm.
and so now we've got this potential treatment for meningitis where by by giving DNases that can break down these neutrophil extracellular traps, you can actually bring back the flow of the lymphatic system
Hmm.
and reduce the swelling on the brain. And they demonstrated it in this model. So it'd be quite interesting to see whether this can translate into humans. Obviously, mice are not the same as humans, so this is still very much in between true and false as to whether it works or not. But
Yeah.
it'll be
And I think that
quite an
the other thing is
interesting, yeah.
what I'm thinking when I hear about this particular intervention too, it's very clear that like this is like in the context of like very sick people that are being managed in the hospital in like some intensive care units, right?
Mm,
Because like you're gonna break apart these nets, but then that's gonna also release all these bacteria into your circulation potentially. But you're probably also getting intravenous antibiotics at this point, right?
yeah,
And so like
yeah,
there's this whole management thing, right? Where in yeah, like it's like it's a way of thinking almost about hospitals that like they just start to take over all of the different functions of your body and just like say like, okay, we know that this is like a disaster, and like, well, how can we just like do different things to fix the problems that are ahead of us so that this particular disaster is resolved, and then we move on to the next phase and trying to like heal up and just let your body like deal with sort of
yeah.
the battlefield that's being left.
Yeah, I think that this is like, because in hospitals you have to deal with like there's prevention of the infection, and there's a point in the infection where the immune system becomes a much bigger problem than the infection itself.
Hmm.
And I feel that that's the stage where this would come in.
Yeah.
Like you wouldn't want this early on, but if it's like at the stage where the immune system in this case is what's causing the brain to be damaged,
Totally.
that's where you'd go, okay, calm down,
Yeah.
immune system, please.
Exactly. Exactly. Cool.
And what they found is that DNase it stops neutrophil extracellular traps, but the neutrophils are still in there. They're still around the brain and doing the other jobs that they're doing.
Mm. Mm.
So, uh, but yeah, no, that's a really fascinating paper.
That is cool. And also like a journal that we don't often read from.
Yeah, what journal is this? Is this I
It was like
oh Annals of Neurology. You're right.
yeah.
Yeah, we'd rarely ever touch this. I mean, back in the days where all you did was like you picked off your specific journal and read from it, we would never have touched Annals of Neurology.
Yeah.
But now, because we have Google, we can now find all these things that are related to
Absolutely.
microbiology.
Absolutely.
Yeah. Next paper is from from a journal we are very familiar with,
Yes.
eLife.
Yeah.
Uh, Evolution of strategy in bacterial warfare via the regulation of bacteriocins and antibiotics. So this paper is to answer a question. So if a bacteria can produce antibiotics with bacteriocins to kill off its competitors,
Hmm.
why doesn't it just do that all the time?
Yes.
Why not always produce all the guns? I mean, it's um, so this is a very mathematical paper where they do lots of differential equations to
Hmm.
try and figure out the game theory of like why do like bacteria like evolve the different ways of producing toxins. So there's constitutive where they produce
It's another model, right?
yeah another
um
model to
maybe maybe like I think it's like I wouldn't think of this like the metabolic model, but you could think of this almost as like the in like some like infection models and stuff. It's like kind of a similar
yeah
idea, right? Because we're trying to say like, you know, if the bacteria are growing at this speed and it takes this much energy to make these types of toxins, and then these toxins have this effect on other bacteria, like who will win in this circumstance? What about if we made the toxins more expensive, right, to create, or less expensive to
yeah
create, more effective or less effective? What would then the balance of power be?
yeah. I mean, we've got all these different systems that have like evolved in bacteria. We've got quorum sensing, we've got sensing of specific nutrients or sensing competing toxins,
Yeah.
and like, well, how does these evolve? And the research bring out the point that okay, let's say you've got like a soil particle that has like a bunch of bacteria on it,
Hmm.
and one of the bacteria goes, I'll just create loads and loads and loads of antibiotic, and at the end of it, it's killed off all of its competitors, but there's less of that bacteria there.
Hmm.
It's won that battle for the soil particle, but it's lost the wider war of being able to spread between different
Yeah.
like soil particles.
Yeah,
And
because it would
so
have
that
invested all of its energy, right, into just making those toxins and have not enough to go somewhere else and replicate itself to enough numbers.
yeah. So the key strategies are not just about killing off competitors, but also dispersing, being able to
Hmm.
disperse and so looking at the wider war rather than each individual battle,
Yeah.
which is a interesting way to look at it. And they talk about how different strategies are releasable, such as toxin sensing, which is almost like a tit for tat strategy where if you sense someone else fighting, you respond in the same way. Um,
Yeah, I love this. I mean, I really enjoy all of these different like social strategies, really, right? Like it's like we're talking about like how do you sense who your competition is, and then how do you deploy some sort of like yeah strategy to deal with that in the most efficient manner possible. It is kind of strange and anthropomorphic sometimes that we're like like we're imagining these as like having these thoughts, but in some ways it's quite apt, right? Because bacteria do have to manage all of these different information streams because they want to be successful, because they want the maximum spread for their cells.
yeah, so it's yeah really fascinating. Yeah, looking at like what how like a game theorist might look at these different strategies. Um,
Hmm.
they and it's also like just got some really cool writing in it that I quite liked. So like they think like they mention like so I don't know if you know about this tit for tat strategy. It's like I think
Yep.
like there's this idea like in certain game theories like doing the prisoner's dilemma that some of the winning
Hmm.
strategies are simply to replicate what your opponent's doing, and then that
Yes.
just forces them to cooperate, and then that's like a a way to live your life, I guess. Um,
Yeah. I mean, in the absence of maybe more nuanced discussions, right? Like that these bacteria can have, like this is what they got, right?
yeah,
They got the bat and they got the way to make the bat. They don't have to make it all the time, but when they do make it, it works.
yeah.
And they know that others can also make their bat, and so there's some middle ground, right, where they can agree to sometimes live together at certain numbers, but maybe when they see their advantage, then they want to push that advantage. That can happen.
Yeah, I mean that's why quorum sensing and sensing nutrients around. So like, do things change completely when the nutrients are low? Then like the knives come out, or like when there's lots of your
Hmm.
people there, and look, that's what quorum sensing is sensing. So yeah, it's and also quorum sensing isn't just you can sense between different species. So it's there's a lot of like interesting like bits to this. Um,
Yeah. And so it seems like the takeaway from this is that they think that one strategy is particularly good at ensuring that they're using their toxins or they're using their bacteriocins or antibiotics in the most efficient way possible while balancing against dispersion.
yeah, they ran this model where they gave these they in their model they gave all these bacteria different strategies,
Hmm.
and they ran a ton of different times in order to figure out which ones tend to win out a lot. So things
Yeah.
like in certain situations down regulating toxin production after a delay, or like
Hmm.
how it responds to different things. And well, they don't so much find one strategy; they find multiple strategies. Um,
Gotcha.
so and some of it is quite like I think situational based. I think maybe, but yeah. But what the key thing they basically say is like regulation is really important. I think
Yes.
that
Yeah.
if you were
You
just a
can't
bacteria
always
that
have it on.
yeah, if you are constantly firing machine gun bullets, you're just going to end up with lots of empty rounds rather than like killing off everybody. So like
Hmm.
so yeah, the idea that bacteria have to be careful about when they apply their tools to fight other bacteria.
Hmm.
Yeah, but yeah,
Cool.
it's a modeling paper, so there is to some
Yeah.
extent like there is always some kind of pre-existing things that go into that. But it's an interesting way to look at it.
Yeah. Yeah, and I just think it's like I'm very fascinated about like yeah, modeling all this behavior of bacteria. It really puts you in the shoes, I think, of
Mm,
like thinking about like these cells because like to model it, like they have to think about like what would the back right, what's the behavior that we're seeing or that's
mm.
possible. Yeah, it's very I don't know, yeah, theoretical behavior actually. It kind of makes me think about and I do love this series, the Foundation series or whatever.
Yeah. Oh, psychohistory.
Yeah, yeah, I feel like this is like psychohistory for bacteria.
Yeah. Well, this is where we
Okay.
start.
Okay. Moving on.
Yeah, moving on. Mutational hotspot that determines highly repeatable evolution can be built and broken by silent genetic changes. Now, this is an interesting one. So what they found was they had this like strain of soil bacteria called Pseudomonas fluorescens that
Hmm.
has a couple of genes that regulate motility. Um,
Hmm.
and what they found was that they're both these two strains are both derived from like the same one, and they lack the function of the master regulator of flagella motility. Um, and both of them can re-evolve flagella mediated motility, so they can both re-evolve it back after it's been deleted. But one of
Yeah.
them like can evolve. One of them finds multiple different ways to re-evolve it. They kind of co-opt different genes. They mutate in a way we'd expect, where it'd be completely random. Another
Yeah.
one finds one way, always that one way, and it gets it back. And they're asking the question, well, why is that? Surely they
Hmm.
should both be random. What is this non-random mutation that keeps occurring, and why does that keep happening? Um,
Yeah. So it's like almost it says like by design or yeah, by
yeah,
evolutionary design, right? There's a section that this is the place that mutations should happen because we will encounter scenarios where it will be beneficial for some people to accumulate a mutation here. Um And so yeah, and so like that becomes that's the hotspot that they're talking about.
Yeah, we always tend to assume that like mutations happen because they're adaptational, but
Yep.
and that eventually they'd find the best way. But
Yep.
in this case, what this paper looks at is it looks at like the mutations, and they kind of go, okay, well, is it they a lot of this paper is actually ruling out the different things that it could be, and they
Hmm.
figure out that it's like there's some silent mutations that don't affect the proteins that are being made. What they do affect is the way the DNA gets tangled up.
Mm.
So there are some mutations in the gene that causes the DNA to get slightly more tangled, and that makes it harder for it to be replicated, and it causes the more errors in that area than you would expect. So
Oh,
that
that's
becomes
interesting. Yeah.
much more
The shape.
susceptible to mutation.
Hmm.
Yeah,
Because
it's
sometimes when people talk about hotspots, the first thing that this reminds me of is that like there are like phage integration sites are
right.
sometimes hotspot areas. So like it'll be like the phage can jump out or the phage can jump in, and that's the switch to turn on or off that phenotype. But here we're saying that yeah, there's also there's another way that that could happen that a hotspot could be generated is that accessibility of the particular DNA strand, and that there are mutations that could be made to make that easier or harder to come about.
Yeah, like so essentially like what they're saying is like when the DNA polymerase enzyme runs across the strand to copy the gene, some areas of the strand are just so tangled up that it can cause it to skip or cause errors to
Hmm.
cause a mutation, and that and these are silent mutations, so they don't actually make any difference to the protein being made, but it's just the way
Right.
that the codons are created. So just yet another one thing to think about um, when looking at DNA mutations. Um,
Yeah. And
so
just thinking about, I think also thinking about the role that a genome plays, not just in the information that it contains, but its physical, right? Its physical orientation.
yeah,
Yeah.
apparently there's a region where the DNA strand looped back on itself, forming a hairpin shaped triangle, which
Hmm.
could cause some issues in the way it was read out. Um,
Yeah.
yeah, so sometimes mutations happen not because they're adaptational, sometimes because the DNA strand literally just is broken in that certain way.
But no, but it is
Oh,
adaptational here in this
yeah,
circumstance, right? Yeah. So
yeah,
like yeah, I guess like what are we saying? It's like the because what when we talk about a hotspot, we're saying that there's an advantage to having this able to be turned on and turned off.
mm,
And then here we're saying like what's the mechanism of opening up that hotspot? And
yeah,
as I said, I think of it as maybe being phage, or like that's one. But like here in this paper, they're talking about like the structure of the genome, the physical structure of the genome as driving those changes.
yeah,
And
and
so then
yeah,
so you do get selection. You get selection for these silent changes, right? That make this region the hotspot.
yeah. I guess, but I mean, the question is the process where those. I mean, because yeah, because this hotspot, in order to be like propagated, has to be like replicated like 100% well, which is quite. Yeah, this raises a lot of like interesting questions.
I it also it the other thing the other story that this paper reminds me of is the fact that Pseudomonas infections inside of cystic fibrosis lungs
Mm.
undergo like their own evolutionary trajectory, and they go from modal to non-modal. It's funny to
Yeah,
think that yeah, you also want to be able to switch
yeah.
back. If you're living in the soil, you want to be able to switch back because like you just don't want to settle down. But and maybe that's true
That
even for the ones that go in the suit in the lungs of CF individuals. I'm not sure.
I mean, that could be an interesting idea. The idea that there is a mutational switch that is regulated by the actual structure of the DNA.
Yeah,
And I
yeah.
guess that'd be quite useful. Because again, I was thinking like bioengineering, like what more tools could we use? And here's another one.
Yeah. I think that the hotspots are useful in like I think of hotspots as being adaptive at some level inside of a genome because like the outcome you only want to survive,
Mm,
right? And like you just don't know all of this is, it's going back to what we just talked about in the competition one, right? Like you don't know like when you'll need a certain set of your genes and when like it's time to move on, right? You've dispersed into a place where you never need them again, and so they should just become relics of the past. Like what I think is being observed in this particular instance is like the genes are still useful in some circumstances, right? The bacteria may still encounter the scenarios where these genes are useful, but they are also very expensive to operate. And so making sure that your population has a mix of individuals that can and can't use it is the strategy for survival, right? Because in the absence, I guess, it could be in the absence too of more formal regulation in terms of like a master regulator, the two-component regulating system or something like that. Like this is a form of regulation just at the population level.
Yeah, I like that idea because I think we see that in other like bacteria as well, where there are like mutations that switch to hypervirulence that seem to operate at like a population level so they're adaptive but only at a. So yeah, it's interesting.
Yeah, and that's also related to like the pseudovirus idea, right? Where viruses like you get and we've seen that in SARS-CoV-2, right? Like you get all this inter-individual variation, intra-individual variation, right? Where it's like the variation there might be doing something, but only a few get out in the transmission bottleneck. But that's part of the life cycle.
Yeah.
Yeah. Okay. And the last thing that we have to share with you guys is de novo biosynthesis of bioactive isoflavonoids by engineered yeast cell factories. And this really is these types of papers are like sort of at the top of the cutting edge, we'll say, of synthetic biology and engineering, right? Biological engineering, where we have molecules that we want that we want to make, and we have organisms like yeast that we are intimately familiar with, all of the different pathways and things that go on inside of them. And in this particular paper, the authors are trying to create isoflavonoids. And they do so by engineering a yeast to do it. And that is a very involved process because this molecule is quite a complicated structure. And you don't want to have to provide a complicated starting reagent, so you have to have all of these different steps inside of the one yeast, and you don't know if that yeast is even going to like having all of those steps replicated inside of it. And this is basically a paper summarizing the work that went into making sure that they could put this pathway into the yeast, and that any complaints that the yeast is raising for having this synthetic pathway inside of it are being addressed.
Yeah, they take all these like enzymes from, because usually these isoflavonoids are produced by plants, and they took a bunch of plant enzymes and they tried to rebuild that same pathway inside the yeast, and then they did a bunch of like work to, like, as you said, try and make sure the yeast doesn't spit it out and doesn't like get upset too much by everything.
Yeah, and isoflavonoids, I think people eat them as like antioxidants, like they're definitely vitamins. But it's also like the base of that molecule is like super useful for synthesizing a variety of different things. So that's like, there could be pharmaceuticals, there's like just many important industrial sort of processes. And so being able to make it in yeast instead of having to purify it from lots of plant matter, it just would introduce like a lot of efficiencies into the industrial process.
And like once we have that more control over things, it means that we can add in and take out extra enzymes depending on what product we want. So there's a lot of
and so what I really like, again, like we haven't really covered a paper like this. This might be something, if people are interested, we'll go into more detail, but just like what are all the problems that can occur, right? Like we know that we have this understanding of metabolism, right? We have a map, right? We saw it in the mosquito one, right? Like you can put this in a computer, right? And you can predict things that happen. But then when you try, and you might think, people might think that because we can predict it in a computer, then we can just cut and paste it from one organism into another, and then we'll have, working, right? This is genetic engineering at its finest, right? Just cutting and pasting genes into the place where they work. But like there is this whole, all these interactions that we could never predict, right? Like through that cut and paste action, there's this different flexibilities in the substrates things use, incompatibilities, even just like making more of a certain compound might all of a sudden show that another enzyme also can use that compound, just like when the concentration reaches high enough. And these are like the problems that you encounter when you do this type of engineering. And so in this particular one, they do actually encounter these problems, and they show how they discovered it and how they engineered around those problems. Yeah, so they specifically talk about, like I think some redox issues, right? Where like there's there's some compounds that are, I guess like energetically favorable to go down a certain path, and they need to prevent it from going down that path and instead go down the preferred path. So they have to add blocks and things into certain pathways.
Oh right, yeah, no, this is really intense. And it does need that deep background in yeast biology in order to figure out like how to get these to work.
Yeah, like I don't think I could. Yeah, this this is sort of it's a very engineering thing, right?
Yeah.
Like you do the thing and then you just try to fix it. Like they did the cut and paste and then they're like, oops, a problem occurred. How do we bypass this? How do we add in some other proteins or remove some proteins in order to make sure that this can move forward?
Yeah, no, that's the thing that strikes me. This does feel very engineering in terms of like testing the process and making sure things flow, troubleshooting. It's
troubleshooting. Yeah. It's a big troubleshooting paper in many ways, right? Like, how do you make this happen?
Yeah, and in some ways, I think that's more useful if you're working in the lab than if you just have a paper where everything goes right, because then it gives you more stuff to look out for, more like, because
yeah, well, I mean, we talk about. I mean, I still am incredibly excited about sort of discovery style papers where we're uncovering, right, some new thing that exists in the world, but I think more and more, I mean, a lot of what biotech is these days is like, how do you troubleshoot biological systems? And I think it's interesting. You can learn a lot. You can also, in some ways, I don't know a lot about yeast biology, but if I went through and I read this paper to try to understand how they did it, I would just end up learning a bunch about yeast because it's building on that knowledge. Without that, without all of those, yeast biologists in the past trying to uncover the mysteries of cell division and like, and different biosynthetic pathways in yeast using plethora of mutants, like we wouldn't have the knowledge to even begin troubleshooting these types of processes.
Yeah, for sure. This is I'm really optimistic for the future of like biosynthesis and yeast, and we'll get all sorts of interesting stuff like coming to. Yeah, this is gonna be really cool.
Yeah, I mean that's the hope, right? The hope is that, and they do make the argument in this paper as well that like the previous industrial way of making isoflavonoids requires all these solvents to dissolve the isoflavonoids away from the matter, right? The biomass that they're purifying it from. But what creating it in a yeast gives us is a more concentrated form that maybe we don't have to use all those solvents, and so maybe it would be more green. I mean, it doesn't guarantee that it would be, right? Like that's just like there's a lot of other technology that has to go into thinking through those issues, but the potential is there, and I think that that's what people are excited about. In general.
I mean, I'm predicting like T minus like maybe a year until someone plugs this through an AI to try and make it do. Because this is,
no, I think that we'll see. I think we've seen. I think it is being used already. I mean, in some ways, we've talked on this program about how like research gets hidden. Especially when like companies start doing them and they're like pushing for technology, they only get shown like when regulatory bodies want to see it. And I do think that that's something to be concerned about in the biotech field, right? Is that like how much of this stuff is going to be hidden, right? Like that AI that's being deployed to optimize metabolic pathways, that is a tool for a company to own, right? To sell and make profit off of versus a research thing that's available to everyone so that you and your favorite system can optimize something. I think these are access issues with technology that I'm particularly interested in learning more about and figuring out like the best practices, right, to make sure that you don't get technologies that exacerbate inequalities, which is such a tricky thing to figure out, I think.
Yeah, I think that is a note to bring our show to a close for today.
Yeah, absolutely. Join us next week for more cool microbiology news. We may actually be ready to do our deep dive by then. So just follow us on Twitter. We'll
Yeah,
make an
we'll
announcement.
we'll update you. We're putting things together. We're trying to do it as best as we can. We really want this one to sing. Yeah, and we want to remind everyone that while we're very, very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can also reach out to us over Twitter at microtwjc. We both believe that peer review is a process and that we can all participate in it. So we hope you've had a good time listening to us ramble on about microbiology today. If you think you have something to add or found something unclear, just let us know.
It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content. Bye.